Alright, so next up, we've got this question which says, the human liver contains a high concentration of enzymes called transaminases. These enzymes are important in the metabolism of amino acids. Amino acids are absorbed from the alimentary canal and carried to the liver by a blood vessel. Now the part B states that there are different types of transaminase. Each type has a specific active site, and they're asking us to explain why this means that each type catalyzes a specific reaction. Now, according to the mark scheme, there are 5 possible points listed, and you just need to mention any 3 of them to get full marks. So, we write, each type of transaminase enzyme has a specific active site with a unique shape, like a lock in the lock and key model. The shape of the enzyme's active site is complementary to the shape of a particular amino acid, meaning it fits or matches exactly. When the shapes match, the substrate, which is amino acid, binds to the enzyme, forming an enzyme-substrate complex. However, if a substrate has the wrong shape, it cannot fit into the active site, so no binding occurs and the enzyme cannot catalyze the reaction. This is why each type of transaminase only catalyzes one specific reaction. That explanation clearly shows why the specificity of the active site means each enzyme only works on one reaction. Then we have another paper 2 question which says, there are many different types of amino acids. One of these is phenylalanine. The diagram shows the structure of a molecule of phenylalanine, and with that, there's a diagram showing the chemical structure of phenylalanine. Now, the first part of the question tells us, phenylalanine is metabolized by an enzyme with the name PAH. And it asks us to name the human organ that metabolizes amino acids such as phenylalanine. This one's just a 1-mark question, so we'll simply write liver, because the liver is the organ that metabolizes amino acids. Amino acids are carried to the liver, and based on general knowledge of amino acid metabolism, the correct answer is the liver. It's the primary site where this process takes place. Then, in the second part, they ask us to explain how the shape of PAH enzyme molecule is important to enable phenylalanine to be metabolized. This part is worth 3 marks, so we'll need to give a full explanation. According to the marking scheme, there are 4 key points, and you usually need to include any 3 of them to get full marks. So, we'll write something like this. The shape of the PAH enzyme molecule is important because it has a specific active site. This active site has a complementary shape to the phenylalanine molecule, allowing the phenylalanine to bind to it precisely. This binding forms an enzyme-substrate complex, which is a necessary step for the PAH enzyme to catalyze the metabolism of phenylalanine. That clearly explains the role of enzyme shape and binding in the metabolic process. Next, we have another question from the same paper. This is question 7, which says, outline the importance of the following in seed germination. Now, we already covered the cell division part earlier when we were practicing past paper questions from the previous chapter. And we had left enzyme to be done with this current chapter, so that's what we're focusing on now. We need to explain the role and importance of enzymes in seed germination. So, we will simply write, during seed germination, enzymes play a vital role. The enzyme amylase breaks down starch stored in the seed into glucose. This glucose is used in respiration, where energy is released. Other enzymes are involved in respiration to help release this energy. The energy produced is then used for protein synthesis and other growth processes. Without enzymes, these essential steps in germination would not occur efficiently. That's a complete explanation that clearly shows how enzymes are essential at every step during seed germination. Then, we have an MCQ which says, a dilute starch solution turns blue when very dilute iodine solution is added. This color disappears as the starch is broken down by amylase. Along with that, there's a graph shown. The graph displays the disappearance of the blue color at different temperatures. It plots time for blue color to disappear in minutes, on the y-axis, and temperature degrees Celsius, on the x-axis. The shape of the curve is a U-shape, with the minimum point at around 40 degrees Celsius. At lower temperatures like 10 degrees Celsius and higher ones like 60 degrees Celsius, the reaction takes a longer time. The shortest time, meaning the fastest reaction, is observed around 40 degrees Celsius. Now, our task is to pick, out of all the given options, which statement explains the shape of the curve. And the correct answer here is B. Enzymes have an optimum temperature range for activity. Why? Well, the graph shows that the time it takes for the reaction to occur is shortest at around 40 degrees Celsius. That's where the enzyme is working the fastest. As the temperature goes below or above that point, the reaction slows down, taking longer, which tells us the enzyme activity is reduced. This kind of pattern, where there's a peak in enzyme activity at a certain temperature and it drops off on either side, is exactly what we'd expect when enzymes have an optimum temperature range for activity. Alright, so then we have another MCQ, and this is a tricky one. It says, amylase is an enzyme that breaks down starch to maltose. Students set up an experiment to investigate the effect of different temperatures on the action of amylase on starch solution. Then they measured the amount of starch remaining after 30 minutes at different temperatures. Which graph would you expect the students to draw from their results? Alright, so we know that amylase is an enzyme that breaks down starch into maltose. In this experiment, students added amylase to a starch solution at different temperatures and waited 30 minutes. Then, they measured how much starch was still left. Now here's the key point. If the enzyme worked well, a lot of starch was broken down, so less starch remained. If it didn't work well, maybe because the temperature was too low or too high, then more starch remained. So when we look at the graphs, we're actually looking for the lowest starch amount, because that tells us when the enzyme worked the best. And in terms of temperature, low temperatures means that enzyme works slowly, so more starch is left. Around 37 to 40 degrees Celsius equals enzyme works best, so least starch left. High temperatures means enzyme gets denatured, so more starch left again. So the correct graph should show a dip in the middle, where the starch is lowest, and high levels on both sides, where enzyme activity is low. So, now let's look at the graphs. Graph A shows starch amount going up with temperature, which doesn't match enzyme behavior. Graph B shows a big hump in the middle, that means most starch is left at the best temperature, which is the opposite of what we expect. But graph C shows high starch remaining at low and high temperatures, and low starch at around 40 degrees Celsius. That matches what we expect, enzyme works best in the middle, and poorly at both ends. And also, graph D is mostly flat. It suggests enzyme activity doesn't change much with temperature, which isn't true. So the correct answer is graph C. It shows the real effect of temperature on enzyme activity based on how much starch is left. And finally, we have an MCQ which says, which statement identifies the optimum temperature for enzyme activity? So, from all the options given, let's break it down. Now, in science, the term optimum refers to the best condition, in this case, temperature, at which a process or organism performs at its best. For enzymes, that means the temperature where the reaction rate is at its highest. And D is the correct answer because it gives the most accurate definition of optimum temperature, the temperature that results in the highest rate of the enzyme-catalyzed reaction. With this last question we have completed our past papers practice for this chapter as well, and with this session sums up our entire Chapter 5 preparation. I'm so happy that we have covered all chapters from 1 to 5 in detail on this channel along with the provided topical notes in the description and pinned comment. For each unit, a playlist is made in my YouTube channel, you can go and check them out. I hope my videos have helped you in your exam preparation, and if you have any queries feel free to ask in the comments below. If you like my videos please do subscribe this channel and like my videos. I'm working very hard to bring each aspect of this syllabus to your site so you don't skip anything and learn with joy, in the highest quality possible. Also fun quizzes are there for your self-assessment, you can check them in the shorts section. Thank you for watching, see you next time, take care and stay tuned.